Hello chaps, I am the renowned scientist Sir Frank Smith, of the University of Astoria, London. I have been given the opportunity to work with the Project DNA Moscow. I have been sent to Russia to work with other scientists, each representing their own nation and the United Nations in this operation. Mr. Brown, the representative for Germany, has informed me that I must do my report for the DNA antisense strand TAC, GGG, CCC, GAT, AAA, ACT, and must present it in front of all of you, the entire group of representatives from the United Nations. Let's get started. You see, our bodies are made up of tiny structures called DNA, which together make up codes to how our body should be made up. Each DNA has a sequence, and it is called a sense strand, and if its sequence is the same as that of the messenger RNA copy that are translated into protein. The sequence of the opposite strand is commonly called the antisense strand. The sense strand corresponding to my antisense strand for my project would be ATG, CCC, GGG, CTA, TTT, TGA. Another major part of the research is the mRNA, or messenger RNA, which is an RNA molecule that encodes a particular chemical blueprint in the protein, which is the case in my case would correspond to AUG, CCC, GGG, CUA, UUU, UGA, and that's it. And the tRNA is another type of RNA molecule that transfers a specific active amino acid to a growing polypeptide chain at the ribosomal site, which would in this case be UAG, GGG, CCC, GAT, AAA, ACU. The corresponding amino acid chain for this would be methionine, proline, glycine, leucine, phenylalanine. The first step is called translation, where there is a copying of the DNA into mRNA in gene expression. This happens in a number of steps and occurs in the cytoplasm of prokaryotic cells and the nucleus of eukaryotic cells. The first step is initiation, or when there is a media of the binding of RNA polymerase and the initiation of transcription. Finally, there is a termination in eukaryotes. This is called polyadenylation, and in prokaryotes the RNA transcription stops when the newly synthesized RNA molecule forms a GcRNA. Transcription stops when the newly synthesized RNA molecule forms a GC-rich hairpin loop following the runs of U's. The second step of the protein synthesis would be translation. Translation is the production of proteins by decoding mRNA produced in transcription, which just happened before the second step. Translation occurs in the cytoplasm where the ribosomes are located. Ribosomes are made of small and large subunits which surround the mRNA. In translation, messenger RNA, or mRNA, is decoded to produce a specific polypeptide according to the rules specified by the genetic code. This uses an mRNA sequence as a template to guide the synthesis of a chain of amino acids that forms a protein. Many types of transcribed RNA, such as transfer RNA or tRNA, ribosomal RNA, and small nuclear RNA, are not necessarily translated into an amino acid sequence. The first step of this is activation, where the amino acids bond with the correct tRNA. Next is initiation, where a small subunit of ribosome connects to the mRNA via initiation factors. Finally is the termination, where the A site of the ribosome runs into the loop of the stop codon. But then a tRNA cannot read this, but a releasing factor then releases the polypeptide chain and the system. This all occurs within the cytoplasm of the cell. Here is a diagram of how transcription works, the three different steps. There is the beginning product and there is the ending product. Here is the different steps of translation, as there are three steps to that as well. Finally, an amino acid chain is formed. In my case, this would be methionine, proline, glycine, leucine, phenylalanine. I thank you for listening, and I do hope that I do better than the next scientist who must prepare his presentation before you.